to have him here, and I'll just say a few things about him and his work. So his academic journey began with a BS and an MS in molecular biology and genetics and biochemistry in Turkey. And then he went to the Max Planck Institute to get a PhD. And since 2018, he's been working in Georgia Tech with the Will Ratcliffe's group there. And he has launched what has become what is known as the multicellularity long-term evolution experiment, or MULTI. That's really the first long-term lab evolution experiment focused on the origins of multicellular life. And I think he's going to talk to us about that today and how you can get that working in a yeast system. Under Ozan's leadership, the experiment has now tracked thousands of generations of what they call the snowflake yeast. And there's really cool properties that have emerged from this system. You can actually see these multicellular yeast clumps with the naked eye. And we've learned a lot about sort of how multicellularity has evolved because of this system. So beyond just sort of the evolution of the size and structure of these clumps of yeast that are like individuals, Ozan and colleagues have uncovered some really fundamental processes like whole genome duplication events and lasting chromosome copy number changes that appear to drive this adaptation to a multicellular life history. So it's really amazing stuff. It seems like they produce a different paper in Nature every year or so. So we're really lucky to have Ozan here today to tell us more about his research. So thanks.
Hello, everyone. So I'm super excited to be here. So one specific reason is that when I was a PhD student back in Max Planck, Germany, we basically came up with this idea of organizing a journal on experimental evolution. At the time, I didn't get on experimental evolution, but the first word that was chosen by me. And the paper was work of Molly's that she did with yeast genetics, and where she evolved yeast for different generations, standing for deliberation. And it was such a pleasure to hear from her, well, 12 years later, that she's also interested in our work. So I'm really happy that I was interested in this year. So I'm going to talk about basically the long-term evolution experiment that I've been doing for years. So the talk has basically 4 different sections. I'll start with a quick background and move on to the results that we've obtained in the last 5 to 6 years. And then I'll move on to more of the recent projects that we are currently running in the lab that are not yet concluded. And finally, so multicellular life is amazing, right? So you see this phylogenetic DNA, it evolved independently more than 45 times in 45, at least 45 independent stages. And here in this tree, everything that is written in bold are a representation of an independent origin, multicellularity. And you'll see the things that you recognize the best as being multicellular as animals and plants, but also it has generated these very interesting, much simpler multicellular structures like C. flexa that has no cellular differentiation, but yet shows certain multicellular behaviors. And then we have, well, for instance, again, small size C. perkins that produces and reproduces similar to our embryo type, but then they split into single cells after a day or 2 days of growth. So there's this huge range of variation in how multicellularity evolves during the history. But despite that differences, we simplify things so that we can study this transition much easier by splitting it into 2 different steps. The first step people studied is where you have started single cell solitary individuals. They form groups, which we call group formation. So they can form groups by aggregating, so different cells come together and adhere to each other, or once cells divide, they don't separate, so they remain together. So that is a different way to produce multicellular. And the second step, in this case, you see cells that are more or less autonomous, so they don't function as a multicellular individual, but then when they undergo through long-term sustained evolution for group level traits, you see them transforming to a multicellular individual so that the single cells now become parts of this integrated whole. So this is basically two steps that we basically break down all these transitions to multicellularity. So when I go over and ask what we know about each step, we know some substantial amount about the first step, group formation. And specifically, groups form for different reasons. And forming a multicellular group is beneficial for different reasons that we can count the top 10 different reasons that are listed here, from things like forming a group protects you from abiotic stress. It's like having multiple cells together shields you from the environmental stress so you can survive harsh conditions. Or this figure that is presented here, you can form this beautiful fruiting body where you can better disperse when the resources are limited, when the food sources are depleted. By forming this fruiting body, you can disperse to different regions. And that is how multicellularity can be beneficial. And the most, basically the popular way why multicellularity is useful is that it allows you to escape from predation. So what you're seeing in this clip is that we have this single cell yeast in red colors. And we also have these clusters of snowflake yeast that are in blue color. And if you look at the rotifers, these are single cell predators. Inside their insects, you're going to see everything is affected. So it can eat up the single cells, whereas the multicellular groups can survive through predation, right? So there are multiple benefits to group formation. And when you look at what we know about the group transformation, though, the way we study it is mostly through the social evolution theory. So we are interested in, for instance, how relatedness is important in forming and transforming a group into a multicellular individual. We look at that when there are cheaters and they spread to a multicellular unit, and then the populations collapse super easily. So there are a lot of theoretical models as to how groups can transform. But one thing about this framework is that you basically, this theory, social evolution theory, does not address the biology of how multicellular traits arise and integrate single cells into multicellular groups. So there's a huge gap in our knowledge as to how the biology changes during this transition to a multicellular individual. And the reason for that is simple. So this is a dynamic process that we need to observe, but everything that happened in way back, right? 150 million years or early in time, and they're buried in fossil record. This means it is really hard to study this as a dynamic process. So the way we study in the lab is as a dynamic process is, like Molly mentioned, we have this yeast system that was developed by Mike Travisano and Radcliffe back in Minnesota more than a decade ago. So they basically took the baker's yeast, a single cell organism, and they applied selection for large size. And in just 2 months, they saw the evolution of these snowflake yeast clusters. And this is an ideal system to study early multicellularity because first, it has this beautiful branching growth structure where you start from a mother cell, and through the branch formation, it generates this flower-like branching structure. And why this is useful? First thing is, if you have a mutation here that I depict with this branch, everything comes from that branch will inherit that mutation, right? So you don't necessarily need to evolve germosome differentiation. After a few rounds of reproduction, these guys are going to be cloned. So this is solving one problem. And secondly, this is quite useful to study early multicellularity because these groups have 3 multicellular life cycles. So the multicellular groups grow large, and there's a physical stress that accumulates within the cluster, and they break apart and produce smaller offspring, which then grow to the inheritance. So there's a free life cycle. And then finally, the simplicity of the system is that you can apply selective pressure for large size very easily just by taking this after 24 hours into a liquid, like you see here, where they settle to the bottom of the tube. So they raise to the bottom. And after 5 minutes or earlier, you kill everything on top. You only transfer the bottom to bottom, right? So necessarily what you get in this condition is that the larger clusters settle faster. And because this cluster size is genetically heritable, you can apply selective pressures for multicellular system. So to bring all the work together in just one slide about the progress we made using studying this as an experimental evolution system, we know a lot about group formation. So we know that when you evolve yeast or improved extracellular polypeptide degradation, they evolve multicellular groups really fast. We have studies with bacteria that show under stress conditions like salt stress, they basically evolve very fastly, these filamentous multicellular bacteria. Or in algae, if you select for algae under predation, they very fastly, in just a couple of hundreds of generations, they evolve these multicellular clusters. So this means that multicellular group formation happens readily, very fast. And we know a lot about this process, but what we don't know is that these groups, when you select them for longer, they don't become much larger. So there is a constraint on multicellular size. So they don't get to anything striking in terms of their phenotypes, like as microscopic clusters, which is what Will and Mike saw when they evolved snowflakes for over a year. And also, these groups, when you select them, apply a counter selection, select things from the top, they eventually revert easily back to the cellularity. And more than that, because these transitions, for instance, in snowflakes happen through a single gene genetic mutation, you take a snowflake yeast and pair this gene, pair that portrays mutation, they easily revert back to the cellularity. So this basically tells us that we don't know much about group transformation. We never got to study this second step in our experience, right? So my promise today is that I'm going to show whether we can take, when we apply sustained selection for much longer, can we take this evolutionary results to a step further that can be observed group transformation. And the way we do this is we use this multicellular long-term evolution experiment, in short, we say multi. And in this system, we start with the simple snowflake yeast clusters that are tiny. We have different populations that are undergoing through different metabolic conditions. And basically each treatment, we have 5 independent populations involved in pattern. And we do these transfers every for over years. So to show, basically, I want to show you one of the most interesting results from the first couple of years of this experiment. So to do that, I have a few key questions to ask, right? Specific questions that are important to our system. First, we know simple clusters evolve very easily, but we don't know what selects for larger multicellular clusters. For instance, we don't know whether environmental conditions certain environmental conditions favor larger multicellular size. We have no answer to that. We don't know basically whether yeast that is connected by single cell connections, with deep bonds, are capable of evolving certain biophysical changes that are required to evolve large, strong bodies. The other thing is, we don't know in nature how long it took for macroscopic multicellular to evolve, and whether it was repeatable or not. Does it evolve readily under the right conditions or not? And finally, when you think about the transformation to a new high-level individual. We don't know, for instance, these very simple groups of cells can later evolve simple multicellular traits or not. We've never observed this happening experimentally. And finally, we don't know the genetic changes that are driving such phenotypic changes that is translation to multicellular. So to test all this, I had this very interesting idea during my postdoc. I read about the history of oxygen on our planet. So to summarize this whole 4 billion year history, life starts without oxygen. Then there's an increase in oxygen. There are multiple increases, but one specific increase about 600 million years ago, the oxygen level increases from 1% of what we have today to about 7% of what we have today. Not much, but it's just significant. But when you look at the fossil record before and after this time, what you start seeing is that you see millimeter and centimeter scale macroscopic multicellular clusters appear in the rock. Anywhere you look at the rocks from Australia to China to Canada, you see these macroscopic clusters globally spread across the globe. So people thinking about this, they suggest that there should be a linear interaction. Sorry about this. This is keynote to PowerPoint transition. When you look at this, you see that you expect a linear relationship with increasing oxygen. You should see an increase in multicellular size. So I basically wanted to test this hypothesis for the first time with the snowflake. So to do that, I grew snowflakes either by under intermediate oxygen level, that is 25% of oxygen that we have today. And I also can manipulate the culture to grow them without oxygen and with modern day levels of oxygen. So when you look at the results, whether oxygen has an impact or not, these 150 days and 145 days experiment showed is that from the ancestor, when you grow them under lower intermediate oxygen, there is not much of a significant change in oxygen size. But when you sparge the tubes with oxygen, next thing you see is that they evolve very large size very rapidly. So this is, to me, a summary of my whole postdoc in a single slide. If you have questions, please. I beg you to ask because I didn't. But to cut the story, long story short, is that oxygen is indeed important in the evolution of multicellular cells. And differently from eukaryotes that we see in 600 million years ago, is capable of also growing on without oxygen, right? So if you are obligate eukaryotes using an eukaryotic oxygen, this is the world that it has to be. We need a lot of oxygen. But if you can get away without oxygen using oxygen, that constraint doesn't apply to you. Therefore, you can again evolve large size. So this is an interesting twist that we see in this. So what happens when you do selecting them for much longer? So the result that we see is you look at multicellular size on y-axis and days of evolution on x-axis. The ones that are evolving under limited oxygen, they remain microscopic. They remain around 50 to 100 micron in size. They don't change at all across 10 different populations. But the ones that evolve without the limitation of oxygen, in this case, anaerobic ones, they very rapidly evolve very large size. So this plot is a bit misleading because it's in log scale, so it's hard to make sense out of this significant change. For that, I have to make it more intuitive. I put some pictures. So the first thing is these evolved clusters are now visible to the naked eye. They can reach to 2-3 millimeter in size. And if you basically grow them without shaking, with very high sugar concentration, they can even reach to basically half a centimeter in size. So they can grow very large. And I call them, they evolve this 20,000 times volumetric larger size. I call them macroscopic. And during a presentation I made, somebody asked, what is the limit for something to be called macroscopic? I thought I remember the paper that says 2 millimeters fellow, but I never could find that paper. One thing I could find was this fruit fly, which nobody, would disagree that it is a macroscopic organism, so I killed that fruit fly. It's just they're comparable sizes. I can, easily call macroscopic size. But now the question is to us, how did they hold this large size? Like, what are the changes that are driving this huge phenotypic shift? And for this, I'm going to explain this in different hierarchical levels of organization of life. So we are going to first look at whether there are multicellular traits and what are those multicellular traits. Then we are going to move on to single cell level traits that are driving those multicellular traits. And finally, we are going to zoom into the genomes to look at what are the genetic changes that are driving those phenotypic changes that are observed at different levels of organization. So to go with the multicellular traits, one thing that is interesting is that when you go from something that is microscopic and tiny to something that macroscopic, you need to make significant changes in the organization of cells. So you cannot use the same cell-cell connecting structures to make such large bodies because it requires 10,000-fold stronger bodies to evolve, right? This is similar to basically, if you build a tiny bridge, you can use a wooden structure. And if you want to build something like this, you need to use a different material and you need to engineer it much more different. So you need novelties, right? So similarly, we ask that question in terms of how multicellular snowflake is large size. One thing we know is that because every cell is connected by a single cell-cell bond, the problem is they can only be as strong as the weakest cell-cell connection. So there is a huge problem here. And looking at them under the microscope, we don't reveal much as to how they became evolved to become such strong bodies. And for that, we needed to look at the inside of those clusters. And to do that, we basically took this yeast to a specific electron microscope, which has a diamond blade. So you cut the cluster into slices and take pictures of each of these clusters, which was done by a PhD student that I worked with. And when you bring them together, basically you can build the whole three-dimensional structure. And when we did that, what we saw was there was a lot of breakage. So branches are forming, but they are broken apart. But the thing that keeps them together is that these branches of a single cluster, clonal cluster, are kept together by entanglement. So these branches grow and split into every direction in 3 dimensions, but they are wrapped around each other so that they are basically forming this stronger cluster. So even if you break a single cell-cell connection, they are still stuck together in this entangled large structure. And when you take this microscopic snowflake yeast and apply some stress to test how much stress they are going to resist against, what we saw was they are now 10,000 times stronger. So they basically evolved from something like gelatin to something like wood in terms of how much stress they can take. So that is how much stress they need for them to break. So they became very strong. So to me, these are basic multicellular traits that we looked at. Multicellular size itself is an important trait. Cluster toughness and branch entanglement are the basic changes that are making such strong, tough bodies. And when we ask, what are the cell level changes that are driving this multicellular level changes, we first looked at the cell shape. So it was super obvious. We start with, for instance, 5 different independent populations. This is day 0. And when you look at the cell shape in yeast, it is almost perfect circles. spherical. So if you look at them evolving over time, though, they became much elongated across all different membranes. So there is strong selection that is driving this cell shape change in yeast. And why this is important, we did a model that is packing spheres in 3 dimensions versus packing cells that are more elongated in 3 dimensions. That is, when you elongate cells or when you elongate aspect ratio, what you see is that cells pack much more efficient because packing this sphere, sausage-like cells are much more efficient in 3 dimensions than packing spheres. And this is not only true in a theoretical model. When we look at the cell aspect ratio from our evolution experiment, what we saw was the multicellular size and cellular aspect ratio aligns perfectly linear. So there's a linear correlation. Like more elongated cells give you much larger structures. And the second change that we observed was yeast is normally only reproducing through its poles. So there are certain poles that are marked genetically, North Pole, for instance, and that is the only way an evolved spring can blood film. But when you look at the evolved yeast, they now started to be able to divide through the sides, so what we call side budding. So they are basically using all the space available to bud in all directions, and you can imagine how that helps to the branch and then entanglement. And the final thing is, it's not just the budding angle becomes different, but also the cell-cell connections become much more larger. So this is, we say the chitin that is connecting two cells. The cells are not visible in this picture, but the chitin is very tiny when they're in the ancestral state. But when you look at the evolved one, the chitin structure is becoming much larger. So making this larger also makes them stronger because all cells are. So these are basically driven by longer cells, side budding, and larger bud scars, enabling them to form these very large macroscopic clusters. So what are the genetic changes that are driving these single cell changes? Right? So to do that, it's super easy with yeast. We just sequence the whole genome. And when we sequence the whole genomes, what we found were genes that are in different populations fall into this significantly enriched category that is related to cell cycle delay. So what happens is that when you delay cell cycle in yeast, which means you delay cell division, this spherical cell, because it cannot divide anymore, it starts to elongate towards the poles. So that's why you get more elongated cells. The other way to get this is by filamentous growth. Genes are mutated, so they also generate this elongated cell shape. And finally, cellular budding genes that are the genes that are affecting bud scar size also are affected during evolution. So, but the good thing is you get the genome sequence and you can infer that these might be the drivers of those phenotypic changes. But nice thing with yeast is you can experimentally test it. So what we did is basically we took the wild type snowflake yeast ancestor, which has a microscopic size, and we deleted some of these candidate genes in this ancestor list. So first we deleted genes that make stronger cell-cell connections. That is showed up in one of our sequence populations. You get 40% larger clusters. When you delete one gene that makes cells more elongated, you instantly get almost threefold larger clusters, not clusters. And when you merge these together, these mutations together in the same genetic background, you even get 6 times much larger in terms of 2-dimensional space. So these genes are important in affecting multicellular size. But as I say, the size is on average 20,000 times in low. So this wouldn't explain all the change in multicellular size, right? So one thing that showed up interestingly was that normally we start this experiment with diploids. So yeast has 16 chromosomes, and when it's diploid, each chromosome is found as a set of two, so you have 16 times 2 chromosomes, and that is how we study. So you can see it's in this plot. On the right, we show the copy number. The blue is 2 copies. That is what we have in ancestor. T0 is day 0. But if you look at different populations and different chromosomes, in all populations, what we saw, even in day 200, these are different time points. They almost all of them involve these tetraploid genomes. So they all convergently involve the same whole genome duplication. And I'm not going to get into the detail. Again, this is a longer, but to summarize it, when you have a whole genome duplication in yeast, you move from diploid to tetraploid, what happens is that cell size increases and cell aspect ratio increases, which are the things that are also impacting the cell size, right? But more crazy than tetraploid was over time, they evolved sustained unemployment. So some chromosomes became 6, 7, 8 copies. So there's a huge unemployment evolving in first time, and those unemployees are sustained across different time. So they are quite interesting, but why are they related to phenotype, right? So we found this interesting result. Bryson is going to be, you already know about this, but we see very similar results with Bryson. So what we saw was in 1 out of 1,000 times, normally snowflakes, macroscopic snowflakes make this distinct donut-shaped colony morphology. So when you grow them, they reach to this millimeter scale size. Sometimes they have formed these spread colonies, 1 out of 10,000 times. So they have this mutation. So when you look at this different colony morphologies of yeast, there is macroscopic and microscopic. The main difference you see in the genomes is that there are huge losses of certain chromosomes, like chromosome 8, chromosome 14. They are decreasing in number in almost all of these tested genomes and phenotypes. So it is basically showing us that these genomes are needed to be found in high copy or low copy for their benefits in making them much more large in terms of multicellular size, because they also affect cell size. So bringing all this together, we have longer cells, side budding that allows them to explore 3-dimensional space, stronger cell cell connection, more elastic cells, all are driven by signal nucleotide mutations and also chromosome copy number changes. And to look at one of a few of results from most recent times, I want to go back to this field. So I promised that we're going to talk about group transformation, right? A group of cells become a new multicellular individual, so they integrate, so they become part of a larger multicellular individual. So I showed that they evolve macroscopic cells readily with these changes, but I haven't shown whether they show signs of transformation to a new multicellular individual. And for that, basically, the question is, if these adaptations are truly multicellular, they might have a detrimental effect at the level of single cells. So there is a trade-off idea that we discuss in literature, that is, when we evolve certain multicellular traits, they also lose the ability that decrease the fitness of individual cells. So do we see anything in sign of this, right? So do single cells become less and less because they show these adaptive changes? Secondly, does the multicellular state become entrenched so that reversion back to unicellularity becomes harder? And then finally, do single cells forming multicellular groups show early signs of distinct cell types? So that are depicted in different colors in the last video. So that those are basically hallmarks of this transition or group transformation to a monster. So I'll show some preliminary data look answering these questions. So the first one is what we saw is pretty simple. The ancestral cell is spherical and they are tiny. And when you look at these spindles that are formed between two dividing nuclei, what you see is that they are short. But when they evolve from the cells, these spindles are becoming much and much longer. So what is happening here is that they start to divide take much more longer time to divide. So these cells are now dividing 30, 40 minutes longer, taking 30, 40 minutes longer to do. That means their reproductive rate is declining significantly. They are dividing much more soon. And related to this, when you look at the mutations effects on single cells from previous work, what you see is that most of the mutations that we see are shown to decrease single cell level fitness. So they are actually detrimental to the single cell level fitness reproduction. And the final interesting thing is that tetraploides evolution is also pretty interesting because we have previous work done by different labs, Seriotto's lab, for instance. In yeast, the stable state is diploid. So when you do experimental evolution work with single cell yeast, if you start with a haploid genome, they all end up diploid. If you start with a diploid genome, because it's the evolutionary more stable state, fitness level is higher, what you see is diploid remain diploid. And if you start with tetraploids, they also very fastly evolving genome size. And what this shows is that the tetraploid that evolved in our yeast is quite uncommon to a mouse cell, quite uncommon in a unicellular group. So it is very unique and probably not favored by single cell just because it doesn't provide much benefits to single cells. So the second thing is entrenchment. Previously, I said, when people tested yeast to evolve multicellular clusters, one drawback is that they revert back to unicellular super yeast when you select them for small size. So it happens 20 and 30 days later, revert back. And finally, this is also observed across the evolution of multicellularity in fungi, where you have the instances of fungi evolving multicellular groups, 8 to 12 independent times. But in 2 of those cases, they revert back to unicellularity, which is pretty interesting because they haven't accumulated changes. So the reversion back to unicellularity is pretty easy. And we can actually test whether our yeasts are evolving entrenchment because of this figure I showed, right? So normally they evolve in clusters by a single gene mutation, single mutation in the ACE2 gene. So when you put back ACE2, they usually revert back to unicellular yeast. So I tested whether our more evolved yeast would revert back to unicellular with this single genetic chain. And to do that, as a control, we chose this microscopic yeast that evolved for 600 days. So they also evolved same amount of time, 3,000 generations. So when you take this yeast from different time points and insert these 2 back, they easily revert back to unicellularity. So what happens when you do the same thing with the macroscopic yeast? I'm going to show one example, but this applies to almost all of them. What you see is that in macroscopic yeast, the first 200 days, you put it back, they revert, but when you put it back to more evolved macroscopic yeast, now they don't revert back to unicellularity. So the genetic path back to unicellularity is becoming tougher and tougher to achieve. So therefore, there are more entrenched changes that are making harder to revert back to unicellularity. And the final thing is, we've been discussing about this, because Bryson also wants to do single cell transcriptomics, is that whether they evolve different cell states, right? Is there a signature of different cell states? So this is quite novel. So we haven't tested this in depth, but what you can do with yeast is that you can take a multi-cell cluster, you can break it apart to single cells, and you can sequence the transcriptomes of each cell, right? So then you can gather these single cell transcriptomic profiles to see if they are expressing different genes in a cluster, to see if there are signs of different transcriptomic states as a sign of different cells. So we did this for one population, and we did this by grabbing yeast evolved across 1,000 days from different time points. We broke them apart to single cells, and when we did the transcriptomic profiling, what we see is that it's a multi-dimensional plot. So each dot in this plot shows a single cell's transcriptomic state, and if a cell has a similar transcriptomic profile expressing similar genes, they map on the same similar position. So this is what you see for the ancestor. But when you look at 1,000-day evolved multi-cell cluster, what you see is that they have a much wider variation in transcriptome profile, and they actually map to these 3 distinct clusters that are either expressing genes that are important for cell wall related products, or expressing genes that are important for programmed cell death, or expressing genes that are important for yeast when they rapidly grow. So there seems to be an emergence of different transcriptomic profiles. And to give you some more detailed info about these different states, when you look at, for instance, a set of genes that are picked, what you're going to see is that in one of them that is growing perfectly rapidly, you see the overexpression of lysosomal genes. And for the ones that are doing increased cell wall biogenesis, so you see an increased expression of genes for cell wall biogenesis. And finally, the ones that are on stress response, we see the overexpression of cell genes in the lower class. Showing basically there seems to be signatures of the. So bringing all this together to summarize all the results that we have. So one thing is oxygen is an important environmental factor that is driving and permitting the evolution of larger molecular size. So we need to think about the environmental drivers of these phenotypic changes are particularly important, not just here, but also for the sort of multicellular life on the planet. And we see that they can rapidly and convergently evolve macroscopic size in just 3,000 generations. So in nature, you can assume this transition from tiny microscopic state to macroscopic state can happen very rapidly and easily. And you also see prior to evolving in controlled developmental program, organisms can find solutions to evolve strong, large multicellular structures by innovating the way that cells interact. And these are driven by single cell level changes that are quite unique to yeast that may lead to differences in transcriptome profiles, which then basically make reversion back to the cell less and less achievable. And to bring this back to this image again, about group formation and group transformation, what we basically see in this experiment is that there are definitely signs of group transformation. And for that, because unicellular yeast converges at diploidy, not tetraploidy, the way they evolve tetraploidy is very unusual. And the way they accumulate those mutations that are detrimental to single cells are also quite unlikely. And the reason that is happening is because of the selection at the group level are favoring these traits that are detrimental to lower level parts. So when you revert them back to unicellular, they are going to be quite disadvantageous. They're going to be under quite low fitness. And unicellular yeast basically don't evolve such elongated cells, and they don't evolve cycling like large blood scars again for the reasons that they are detrimental to single cells. So by selecting these multicellular groups, eventually they're evolving a sort of entrenchment so that the reversion back will be observed. And with that, it's been a quite intense and quite fun day here at the integrated biology department. And I really enjoyed my time here. So I'd like to thank Molly and Trista and Bryson and everybody who I talked to for inviting me. And there are many people are now working on this project, so I wanted to highlight it here as well. So that anybody is interested in and have different ideas, we are quite open to sharing this space, which is also part of our statement when I got this NSF Alfred fellowship that is supporting any long-term evolution experiments. So we're quite excited that we are able to share this space. So please, if you have any ideas, let us do so. Thank you.
All right, well, we have a few minutes for questions.
Yeah, great talk. Thank you. So the single cell RNA-seq, I was like, he's got to be doing that. He's got to be doing that. And sure enough, he did. It's awesome. So to me, the next step would be evidence that single cell RNA-seq can be mapped somehow spatially. And so have you thought about that? Like my hypothesis would be the growth would be on the tips and the stress would be in the middle.
Right.
Yes.
Exactly my hypothesis.
Yeah.
We have a student now developing markers to tag just these specific genes that are expressed differentially to visualize where they're overexpressed, to find whether there's a special pattern. So that is one thing. The other thing is related to what you said, for instance. We have stress, right? Some cells are super stressed, and those cells are the ones that are red, which means they're dying, they're apoptotic, and they are more central. So there seems to be sort of spatial structure in terms of how they look, in terms of phenotype. Now we need to map, like I say, that phenotype to
Very cool.
transcriptome and find out why they're. It's the other question, why they're beneficial.
Yeah, great talk. I'm curious about, so the timeline to entrenchment, you kind of took like half the way through for that to be established. Some might predict that epistasis might set on earlier than that and cause kind of fixed changes, but you see this really kind of delayed pattern. I'm curious if you have hypotheses on the reasons why. Something to do with ploidy or I don't know what.
Exactly. So one thing is, first thing we thought was ploidy might be a pattern. That is, we now have 4 copies of each chromosome, and when you replace ACE2 to see if they're going to revert, you're only introducing it to a single point. So we tested that by introducing ACE2 tetraploid is from synthetically developed tetraploid, they revert back to unicellular. So that wasn't a problem, but like you said, one thing we start to see is that the time they start not reverting is when they accumulate these mutations like in genes like gene4 that are making cell-cell connections stronger. There are some mutations that are affecting cell shape. So we are trying to see when we insert those additional mutations and repair them in these macroscopic clusters where they are going to be able to revert back or not. So we are basically functionally trying to test whether we can revert them, putting those genes that are known to be important in cell conditions.
Sense. Yep.
I had a question about the ploidy actually as well. Why does chromosome 8 seem to be so subject to wild swings of ploidy?
Yeah.
This one, I think, and the previous figure too showed like really high copy numbers.
Yes. So one thing is, when you look at different papers, they find out chromosome 8, chromosome 16 are to be important in generating this more elongated cell shape, and this donut shape colony morphologies. And they're basically stating that copy number itself is not that important. So that you might think that all the genes in that chromosome might be relevant to this phenotype. But what people find is that there's certain genes that are super prone to changes in terms of expression when they change the copy. So they are very sensitive to dosage. So we think there might be certain genes that are basically driving this change, not the whole chromosome copy number. So you can still map them to single or multi-figures and see whether they can be driving this quality level changes that are also in the cell shape. But it's a huge project, and we have people interested in knowledge with working.
Perhaps I misunderstood you. The donut monophenotype that you were observing, was that stable? Like if you took a donut out of the tube and it's on a tube, you get more donuts?
Yeah,
You could.
so you can get from a donut, you get 999 donuts plus one spread.
Okay.
So that's how it works. Like it's a random mutation, and it's most likely those unemployed chromosomes are lost. During segregation, therefore you get those different rates, a correlate because chromosome copy changes.
Okay, cool. That's really fun.
Yeah. It was hard work by science and this to improve. Basically spread them into single colonies again, in the sequence of the whole spread.
Yeah, a lot of effort.
Yeah, so I was curious about in terms of the conditions that allow for this to exaggerated. Has there been any thought on like the actual energetic demands of these as they're saying increase in size? So like as they're sort of getting this, are they also having like are things into the future? Like greater metabolism, like the role of that sort of.
Yeah, certain things are different, like first of all, what you start to see is they slow down their glycolysis very strangely. So despite that they are growing fermentatively, their uptake of sugars are slowing down. But maybe that is because they are growing much larger sizes, so the diffusion is not as efficient. You think that it can be for a smaller cluster, but also what we interestingly see is that when they form these donut colonies, they generate this liquid flows. So it takes all the dense sugars from the environment, ferment them, produce carbon dioxide, which basically shoots through the top like a chimney. So they evolve this very strange, nutrient flow mechanism. I think that it speeds up uptake of glucose. That is solely driven by dense differences. So that's the basic difference between found. Is that
Yeah, so because I was sort of wondering, yeah, because I was sort of getting it, is there like a limitation or an uptake, or I'm assuming it's sort of highly saturated. Yeah. So even in a low nutrient, would the phenotype still be stable? Same way it's kind of dependent on oxygen. Seems like if you had greater demand or efficiency issues.
like I think if you decrease the concentration of carbon, it is going to negatively impact the size of the fusion for sure. For a strange reason.
Yeah. Very cool.
All right, well, I think that that's our time, so let's thank Ozan again for the. We still might have questions, we're gonna head over in about 15-20 minutes. I have to,